All right. In the last video, we got to know the main monomers that our cells use to build macromolecules. Before we start looking at the polymers themselves or the macromolecules themselves, we need to look at how we go from a monomer to a polymer or a polymer to a monomer, because both of these types of chemical reactions occur in our cells all the time, depending on what we need and where we're at. So, there are two types of chemical reactions we're going to look at. When we have a polymer or a large molecule that gets broken down into smaller subunits, our monomers, this is called decomposition. When we have a bunch of monomers that we put together to make a polymer, this is called synthesis. Synthesis means to make. Now, chemical reactions in general occur whenever two molecules collide with enough force and in the correct orientation to form a bond. That means that there are several things that can influence how fast a reaction occurs. Reaction rates are affected by the concentration of our reactants. In other words, if we only have two molecules in a space, the odds are lower that they will actually run into each other in the right way and at the right speed. But if we have 100 of each of those different molecules in the same space, it's much more likely that two of them are going to collide and form a bond. So the more concentrated something is, the faster the reaction will occur. The next thing that affects the rate of chemical reactions is temperature. This is because every atom, every molecule has a vibration. So if I'm a water molecule and my head is the oxygen and my hands are the hydrogen, there's going to be a vibration like this, all right, as I move around in the space. And when you have two molecules that are vibrating and they bump into each other, they're both going to ricochet off, and now we have molecular movement. Now, the hotter it gets, the faster the molecules move. They move faster and they hit each other with more force, which means we're again more likely to have the type of collision that will lead to the chemical reaction taking place. Now, the last thing that influences reaction rates is catalysts. Now, catalysts exist outside of organic chemistry, outside of living things. But in biology, our catalysts are called enzymes. We're going to talk about those more on the next slide. Catalysts are things that bring the two reactants close together and in the right orientation to make it easier for them to form the bond or do the reaction. So it will drastically speed up the rate of a chemical reaction, but the catalyst never enters into it. It doesn't become part of the bond. Sort of like a matchmaker, right? Matchmaker finds person A and person B and is like, I think you two would make a lovely couple. And it brings them together and they form a lovely couple. And the matchmaker goes on to do it for another couple, right? So they don't become part of the final product. Now, in our cells, these synthesis and decomposition reactions are done by enzymes. Those are our catalysts. Enzymes are usually proteins, almost always. And they speed up the rate of reaction a lot, like millions of times faster than if there were no enzyme present. And just like the definition for any catalyst, enzymes are not changed by the reaction. Enzymes are also substrate specific. The substrate is the thing that the enzyme acts on. So here we have an enzyme called sucrase, and its substrate is sucrose, which is a disaccharide. Now, sucrose fits into the active site of the enzyme. The enzyme helps with the chemistry to break the two of them apart, and now these two monosaccharides diffuse away from the enzyme. If another molecule came over here and tried to fit into sucrase, like two amino acids joined together, nothing would happen. It won't fit. So sucrase only acts on sucrose. And by the way, this naming is pretty consistent across biology. If you see ASE, that means that you've got an enzyme, and it usually has almost the same name as its substrate. So sucrase, sucrose. All right, so enzymes are very important in our cells because they massively speed up the rate of chemical reactions. All right, let's look specifically at how polymers are formed. This chemical reaction is called dehydration synthesis. Now, the word dehydration means to remove water. So if you dehydrate food to store it, you're taking the water out. And synthesis means to make or to build. Now, with any two monomers, it doesn't matter which ones, we always have these hydroxyl groups. And if you hold the two monomers together so that these hydroxyl groups are nearby, the enzyme that does this reaction, and remember they're substrate specific, so the enzyme that does this for monosaccharides is different from the one that does it for amino acids. So whatever enzyme it is, it holds these two monomers so that their hydroxyl groups are close together, and then it takes off the hydroxyl group from one of the monomers, takes the hydrogen off of the hydroxyl group for the other, and these two things combine to form water. So we've removed water from the bond. Now this leaves this carbon without anything to bond with, and it leaves this oxygen without anything to bond with. So because they're sitting next to each other, they form a bond together. So now we have a dimer, or two monomers that are joined together with this oxygen. Okay, so that's dehydration synthesis. The opposite is true for when we want to break a polymer down into monomers. We have, in this case, a dimer, the two monomers joined together by the oxygen. The enzyme that catalyzes this reaction takes a water molecule, splits it into a proton and OH minus hydroxide, and then it breaks the bond right here, and then puts the proton with the oxygen and the hydroxyl group on this carbon. Now, hydrolysis, its name literally means water breaks the bond. Hydro is water, lysis is to break. And it's the exact opposite of what we saw with dehydration synthesis.